Good morning, ladies and gentlemen. Please take a seat and wait until the session begins. Thank you. Good morning, ladies and gentlemen. Today is the third day of the sixth HOPE meeting. Let me introduce the moderator for this session, Professor Haruhiko Shiomi, Professor of Keio University School of Medicine. Professor Shiomi, please.
Good morning. I'm Haru Siomi. I'm very happy to chair this lecture. Dr. Rich Roberts started his scientific career as an organic chemist. However, he soon got interested in restriction enzyme. Actually, he fell in love with restriction enzyme, I suspect. And as many of you know, identification and characterization of restriction enzyme really opened up a new field of biology, so now we call molecular biologies. And he contributed a lot to open the door. And later, he used his favorite restriction enzyme to study arrangement of genes and their transcript, and he found that a gene is split in piece, and therefore a primary transcript of a gene is spliced in the nucleus to produce mature messenger RNA. For this discovery, he shared a Nobel Prize with Phil Sharp in 1993, and just a year before he got the prize, he moved from Cold Spring Harbor to a company named New England Biolabs. And since then, he has worked for the company, and currently he is he's the chief scientific officer, and very importantly, he continues to do basic research in the company. So I am looking forward to your talk. So Rich, please come to the podium.
Okay, well, thank you. What I'm going to do today is to tell you about the work that I do at the moment, because I think it's really exciting. But I'll give you some background as to how I got involved in it, so you can see some ideas about how my scientific career evolved. I'm going to say almost nothing about RNA splicing that I won the Nobel Prize for, because most of my work is just, as was just said, developed from my love of restriction enzymes and restriction systems and basically bacteria. I love bacteria. I find humans and things connected with them way too complicated. I like the simplicity of bacteria. But at the same time, I recognize, bacteria have been on this earth for an awful long time, much longer than we have. And in fact, we are full of bacteria. We're covered in them. And without them, unless we live in isolation, we will die. We really need these bacteria that live with us. In fact, some witty person once said that perhaps the bacteria invented us so they would have a nice home. So I'm going to talk about bacterial methylomes, but I'm going to start off by telling you how I got interested in this whole subject. And it's really due to one man shown on this slide, the man on the extreme right, Dan Nathans. Now, Dan Nathans was a professor at Johns Hopkins University. He worked in a lab right next door to Ham Smith, who's shown here in the center part. And Ham discovered the first so-called type 2 restriction enzyme. And the very first restriction enzyme, and actually the theoretical basis for restriction and modification, was all worked out by Werner Aber, who working first in Geneva and now working in Basel. And these three shared the Nobel Prize in 1978 for this discovery of restriction enzymes. But my story starts in 1971 when I was a postdoc at Harvard, and I went over to the medical school and listened to a seminar by Dan Nathans. And Dan showed a very beautiful picture that showed that if you took SV40 DNA, a relatively small eukaryotic viral DNA, and cut it with Ham's enzyme, which at the time was called endonuclease R, you got a series of nice bands that were produced on a gel. And at the time, there were no methods for sequencing DNA. Fred Sanger had worked out how to sequence proteins. He'd worked out how to sequence RNA. But the problem with DNA was that there were no small molecules to practice on. Okay, if you want to develop methodology, you have to start off with something simple. And in this case, in the case of DNA, even the smallest DNA molecules known were huge, absolutely enormous. And it occurred to me that these restriction enzymes offered you a way of making small DNA fragments that you could then use in order to develop DNA sequencing methods. And that was why I got interested in this whole field. Now, I ended up not getting involved in DNA sequencing, which was what I had imagined when I moved from Harvard down to Cold Spring Harbor, because as soon as I got down there, I started to make a lot of these restriction enzymes, discovered that the 6 that were known were just the starting point, because everywhere we looked, every bug that we looked at, that we grew in the lab, had a different restriction enzyme in it. And they all recognized different specificities. And I thought that maybe they alone, there would be enough specificities we could sequence DNA with them. But in fact, they had so many other uses, and people were coming to my lab all the time, and they wanted to get samples of these things. And so it was obvious people were really interested in this. And this, of course, was when the biotech industry got started. So it was a very exciting time. I made so many friends at that time because everybody wanted a little free restriction enzyme from me, and we were sending them out and giving them, and everybody loved this. But what I want to do is to back off a little bit and just tell you something about how restriction systems work, how important they are, and some of the fun things that come along with them. So this is a little cartoon showing you the phenomenon of restriction. And so what happens in a bacterial cell that has a restriction system in it is that there is an enzyme represented by this little gremlin down here with a pair of scissors that comes along and recognizes a specific sequence in DNA and then cuts it every time that that sequence occurs. And so if you have a phage infection coming in and you have DNA coming in, this restriction enzyme is able to cut up the phage DNA into small pieces, and that aborts the phage infection. It just can't make all the gene products it needs in order to replicate. Now, of course, the bacterium itself has DNA, and so it has to have some way of protecting its own DNA against the action of the restriction enzyme. And this is accomplished by modification In fact, in most cases, but not all cases, in most cases, this is achieved by DNA methylation, by putting a methyl group onto one of the bases in the sequence that is recognized by the restriction enzyme such that it blocks the action of the restriction enzyme. And then when you have both of these enzymes in a cell, a phage DNA comes in, and it becomes a competition, a race, if you will, between the methylase that is looking to modify the DNA and the restriction enzyme that is looking to cut it up. And nature has basically fixed the game so that the restriction enzyme almost always wins. And it does this by making the restriction enzyme a very good enzyme, catalytically very competent. These things are quite difficult to kill. Many of you probably have them in your freezer. You could probably just leave them out on the bench, and they're usually good for 6 months or a year. It's that they don't have to be kept cold. They retain activity. The methylase, on the other hand, is rather a poor enzyme. It's not very catalytically competent. And if you think about what is needed, it's only during the complete life cycle of one bacterial cell that you need the methylase to have methylated everything during that time, whereas the restriction enzyme has to act immediately. And so nature has fixed the game and the restriction enzyme wins, but not always. Every so often a phage will come in, and maybe it's only got 1 or 2 or 3 sites for a restriction enzyme, and just by chance the methylase gets there first. And when that happens, then the phage is able to grow in the cell, and so it escapes. And so what happens if I put, say, 1 million phage particles in with 1 million bacterial cells, maybe 1 or 2 will grow as phages, and their growth is restricted. You know, most of them die, most of them can't get in, but just 1 or 2 can. And so that's why it's called restriction. It restricts the growth of phage. It's not an absolute immune system. Now, a lot of bacteria get over this by having many restriction systems. In fact, in Helicobacter pylori, which is a bug that is in your gut, or at least certainly all of you who come from developing countries, you can pretty much guarantee you have it in your gut. If you're in a developed country, you probably only have a very low level because we use antibiotics so widely, and we've actually killed most of them off. But in Helicobacter pylori, there are no less than 20 or 25 different restriction systems, all recognizing different sequences that are present. And so it is very, very good at cutting up DNA in phages or in free DNA that gets into the cell. Now, I keep a database of all of these things. This was one of the very earliest databases ever put together. It got started in 1974 for a meeting that I went to in Belgium. Because at the time there were about 30 restriction enzymes, and I thought it would be nice to have a little database where people could go and look and find out what was what. And it just started off as a sheet of paper that we typed out and copied and gave to people. But then later on, eventually, when electronics came along, we were able to produce a proper one, and this is the website that describes this. And if we look at this today, there are actually, if you look at it yesterday, let me go back here. There were 1,934 DNA methyltransferases that had been characterized to some extent. That is to say, we knew that they were active, usually because they accompanied a restriction enzyme, and we also could make a pretty good guess about what the recognition sequence was, although not until very recently have we had really robust methods for working out what the recognition sequences are, and I will get to that in a moment. Now, I spent most of my early years at Cold Spring Harbor studying restriction enzymes, and it was only really after I'd been there for maybe 18, 19 years that we got interested in the DNA methyltransferases. And the reason we got interested in them was several-fold. One is there was no crystal structure for them. And if you look at the sequence of a methyltransferase and a restriction enzyme, we had originally thought that there would be a little section of sequence that would recognize, say, GAA-TTC, and we could work out what was recognizing what just by comparing the 2 gene sequences. Well, we soon discovered, as soon as we started cloning and sequencing these things, that there was just no similarity whatsoever at any point between a restriction enzyme gene and a methylase gene, and that is still true today when we have something like 40 or 50,000 putative enzymes and putative systems showing up. They don't recognize DNA in the same way. They use completely different sequences in order to do so. So we became interested in them, first of all, because they were part of restriction modification systems. We also discovered that they were very easy to find using bioinformatics methods. And the reason for this is that they all interact with S-adenosyl methionine, which is the source of the methyl group, and they have a very common set of motifs that are present that actually do this recognition. So because they have some common catalytic mechanisms associated with them, they do show sequence similarity one to another. And so that can be very useful when you want to find these things if you've just sequenced a bacterial genome or something like that. That is not true for restriction enzymes. Restriction enzymes typically look like nothing other than the restriction enzyme that they are. They typically do not have homologs in GenBank. They're evolving extremely rapidly, and they show very little sequence similarity, even when you find two that recognize exactly the same DNA sequence, or even closely related DNA sequences. Typically, they look very, very different to one another. Now, several of the methyltransferases have shown to have regulatory activity in bacteria. So, for instance, the DAM methylase of E. coli, which recognizes GATC, is known to methylate regions upstream of genes, to overlap with promoters in some case, and they can modulate the transcription of those genes because some polymerases and their associated transcription factors are sensitive to methylation. There's a number of examples of this. The DAM methylase of E. coli is also associated with mismatch repair. It shows which strand should be considered the parental strand, the strand that should be copied when you're making repairs of lesions. And so that's another role of it. It has no counterpart restriction enzyme, so it's only associated with that. They also support epigenetic phenomenon. So for instance, we know that during the cell cycle in Colobacter crescentus and many other organisms, that there is a methylase that recognizes GANTC, and it modulates the cell cycle, and it tells the cell when it should be dividing and when it shouldn't be dividing. So there are a variety of things going on, and I will say more about that towards the end. And finally, they're good examples of proteins that recognize specific DNA sequences. I'm sure you're all aware that there's been a tremendous interest in how proteins recognize DNA. All of the eukaryotic people are very interested in transcription factors. They want to be able to predict what are the recognition sequences of transcription factors, where they bind, and hopefully to then work out regulatory networks. So we were interested in the methylases because they did not seem to have any of the standard ways of recognizing DNA sequences. And so we were kind of curious about just how they did it. And with that in mind, we got started on a project to actually crystallize a DNA methylase, binding to its recognition sequence so that we could see how they did that interaction. Now, before I tell you about those experiments, let me just tell you there are three kinds of methylation that we find in bacteria. So the ones that we find at the top are 5-methyl-C, and this is exactly the same modification that you find in humans, in mice, in higher organisms that are typically associated with epigenetic phenomena. In the prokaryotes, though, many of these enzymes are associated with restriction systems, although there are plenty that are orphan methylases, and typically we don't know what they do. The next kind of methylation is N6-methyl-A, in which the extracyclic amino group over here is methylated. And again, this is very common in bacteria. It's probably the commonest of the methylations that we find. And then occasionally, we find that N4-methyl-C, that cytosine, the extracyclic amino group, is modified. And it's kind of interesting because enzymes that do these two reactions, one recognizing adenine, one recognizing cytosine, cannot be distinguished unambiguously at the moment by bioinformatics. We can make a pretty good guess when we look at a sequence as to what it's going to do, but we cannot yet be absolutely certain of what it's going to do. And one reason for that you'll see in a moment. Now, the other thing that is interesting here is that they have a really interesting mode of interaction with DNA, and that is shown on this slide. And this came from when we were looking at the methylase HHA1, M.HHA1, recognizes GC-GC and forms 5-methylcytosine. We first had a structure just for the enzyme by itself, and we couldn't really tell how it was interacting with DNA. And then, fortunately, just before I had to go to Stockholm to collect my Nobel Prize, we made this discovery that when this protein binds to DNA and is going to methylate the cytosine residue, what it does, it takes the cytosine it's going to methylate and flips it 180 degrees right out of the double helix, right into the active site of the enzyme, where it is available for methylation. And when you think about it, it makes a lot of sense, because normally when these bases are buried inside DNA, they're base pairing, and it is not that easy for enzymes to get at them and do any sort of chemistry on them. Here, we discovered that all of the methyltransferases that we've looked at use exactly this mechanism of flipping the base out in order to modify it. Shortly after that, a number of people showed that the same mechanism was operating for DNA repair enzymes, and then now a whole list of things that use this mechanism to get at the bases in DNA when they want to do some chemistry on them. Now, I say this was a very lucky, timely discovery, because when I went to Stockholm to pick up the prize, normally you give a lecture and you tell people about all this wonderful work you've done on the system for which you won the Nobel Prize. But I'd not worked on splicing for many, many years, and so I was a little unsure about what to talk about, but all of a sudden I had this great discovery. I mean, this had not been predicted, no one was expecting it, and it was a really nice, interesting discovery. And so I used this as the basis for my Nobel talk. And you know, I would advise all of you, if you ever get asked to go to Stockholm, make sure you've got a nice discovery to talk about when you get there. So let's talk a little more detail about some of the systems. There are basically 3 kinds of restriction systems that use methylation for protection. We have the type 1 systems shown up here, and this is a 3-gene system. The blue gene is the DNA methylase, so this is responsible for methylation. The R gene is the brown gene here, is the R gene. This is responsible for restriction for endonuclease activity. But the really key gene is this one, the S gene. This is the specificity subunit because it is this molecule that actually recognizes the DNA sequence and determines whether or not it's going to be methylated or whether it's going to be restricted. So this is the key molecule here. This determines the specificity of the system. And if you want to methylate, you need the M subunit and the S subunit, and just by themselves, they will methylate DNA. And we have a number of cases that we know about in genomes where just these 2 genes are present, and there is no R gene, there is no restriction. So there are a number of cases where you just get methylation of these sequences, but you don't get any restriction. We don't know why that is. We suspect, of course, that there is something interesting going on there, but we don't know what it is. In order to get restriction, you need this 3rd subunit, and you actually form an enzyme complex that has 2 copies of the R gene protein, 2 copies of the M gene, 1 copy of the S gene. Now, determining the recognition sequence for these enzymes is a problem. And the reason it's a problem is that you cannot use the restriction feature to sort of produce nice bands on a gel, because restriction is completely random. It just goes all over the place, and you do not get nice fragments. And so the only way you can get at specificity is by looking to see what are the sequences that are methylated. And this is not straightforward. This is one of those projects that if I came to you 5 years ago and said, hey, you want to come work in my lab? you go and work out the recognition sequence for one of these things. And at first, students say, oh, terrific, yeah, I'd love to do that. And then they read the literature and they discover just how difficult it is to do. Because the way you do it, you have to do methylation of a large set of fragments and then work out exactly what are the sequences that are methylated in a large number of fragments, so you get enough so that statistically you can say, oh yes, this is the core sequence that is always the same. And if you look in this case, you'll see that this actual recognition sequence is composed of 3 specific bases, 6 that can be anything, and then 4 specific bases. So it's not even a nice little sequence that you can get at, and it was a big problem. And you will see when I show you a graph about the discovery of methylases, just how hard it was to do this, because we knew almost no sequences until a couple of years ago. The type 2 enzymes shown down here, these are the ones that you're used to seeing in the lab. These are very nice. You can easily work out recognition sequences because you can take one of these restriction enzymes, cut up DNA, you get a nice fragment pattern, and you can actually analyze those fragments using computer programs and work out what is the likely recognition sequence. Turns out to be a very robust method. We developed it back in 1978. It was one of the very first and what I call interesting bioinformatic programs ever written. Because it did more than just translate a sequence or start to put sequences together and look to see how they overlap. And we still use exactly the same method today. It's very easy to work out the recognition sequences. But you still have the same problem with the methylases. You don't know whether the methylase is going to have exactly the same recognition sequence as the restriction enzyme, or whether it's going to be more degenerate. So for instance, let's say you've got a restriction enzyme recognized as CCGG. If you had a methylase that methylates every CG in the sequence, it will automatically protect against CCGG, as well as TCGA and so on. And so we didn't know in the early days exactly what was being recognized for the reason it's difficult to do, but it was always assumed that the methylase must have the same recognition sequence as the restriction enzyme. And recently we've developed methods and methodology that allow us to show for sure that in almost every case that we've looked at, that is true. Down here are the type 3 restriction enzymes. These are very similar to the type 1s and the type 2s in some ways. They have a methylase gene, they have a restriction enzyme gene. In this case, the specificity is in the methylase gene. And so if you just take the methylase gene, express it, it will methylate DNA. If you want to restrict DNA, though, you need the methylase subunit and the restriction subunit, and then it will cut. These enzymes are like type 2s in some ways. They cut at or close to the recognition sequence, usually 20, 30 bases away, but they have the unfortunate property it's very difficult to get them to cut completely. They like to sort of cut partially. And so then when you do a digest and look on a gel, you get a smear, and it becomes difficult to use the restriction properties to work out what the recognition sequence is. And so these suffered exactly the same problem that the type 1 system did. You had to go after the methylase to work out what was being recognized, and that was a problem. And it was a problem until a little over 2 years ago when a company in California called Pacific Biosciences introduced a method for sequencing DNA that allowed you to not only work out what was the order of bases in the DNA, but it could also tell you when those bases were modified. And so if along the way there was a modified A residue, then the method allowed you to detect the fact that it was methylated. And it goes more or less like this. The polymerase is tethered. You give it a single-stranded substrate, and it works on single molecules. This is a single molecule method. You don't need to clone anything. You can do it directly on DNA that comes straight out of a cell. And it begins to copy. And as it copies, you can measure the time that it takes to add the next base. You've got 4 fluorescently labeled bases. You add them one at a time, and you can see how long it takes to go from one to the other. To start off with, they did an experiment in which they looked at just DNA that came straight out of the cell, and then they looked at DNA that had been PCR'd up to get rid of all of the methylation, and then you can compare the two timings, how long it takes to add the next base. Now we know enough that you don't have to do the unmodified base to do it. You can just look at the sequence as it comes straight out from a piece of DNA that comes out of the cell. And what happens is the polymerase pauses just before it gets to the modified base while it's trying to figure out what to put in. And so the modification just slows the polymerase down temporarily, and then you get a very nice signal. This is just down at the bottom here, looking at the time interval for adding the next base. And so you can see that what happens here, where you've got a modified residue, you get a big signal that is due to the modification, and it tells you that that base is modified. Now, this really looked very nice. We thought it was wonderful when I first heard about it, and so I called them up and I said, hey, we have a very large number of methylases that are cloned. We have them cloned on plasmids. Why don't we try your method and see, does it give the right answer all the time? So we sent them about 20 plasmid, each of which had on them one methylase gene. It was in a strain, it had been grown in a strain of E. coli that had no other methylation whatsoever, so the only methylation was due to this one clone gene, and we knew what the recognition sequence was because we had done that in separate experiments, and in many cases, we even knew what the modification was because that had fallen out from various work that had gone on. So we sent them over, and they ran them through their system, and basically, they got very nice signals. We did, first of all, a whole lot we knew the answer for, and then we sent them some where we knew the recognition sequence, but we didn't know which base was modified. And so in this case, we sent them an enzyme that recognizes AATT, but we didn't know whether it was the first or the second A residue. And here you can see very clearly it's the internal adenine that gets methylated, a very clear signal. And down here, it turns out that it is this first adenine that gets methylated here. But from what we knew, it could have equally been N4-methylcytosine of either of these two cytosines down here. So we did enough of these, looking at known things, at unknown things, the things that look good, that we could be sure that the method that they had developed was actually giving reliable results. And that was really important. So that was very nice at looking at the type 2 systems, and it was that that allowed us to look at enough genomic DNA to see that the specificity of the methylase almost always matched that of the restriction enzyme. Now you might think about that. It's quite clear that if you've got a restriction enzyme, you have a strong selection for a methylase that will methylate all the base, or at least the appropriate base within that sequence. But how do you select for the specificity of the methylase? That is an unknown problem. We don't know why that happens, but it does seem to be happening. So there's probably some good biological selection there, but we don't know what it is. Now we thought, well, this was all terrific, but what about the type 1 and the type 3 systems? Because these are the things we usually had no idea what they were recognizing, and yet we knew that there were a lot of these present in genomes. And so we cloned out one or two that we knew, we cloned out a bunch that we didn't know, and then sent them over to PacBio. And so in this particular case, we knew the answer. It's from an E. coli strain. They gave very nice results. We got the specificity immediately. Down here, this is one we didn't know what it was ahead of time, but this was the pattern that we got. So the little bars on the outside here show the sites that are methylated. When you look at all the sites, the common sequence is CGAAG. It is the second A residue in the sequence that gets methylated. But I would have you notice that on this Circos plot here, the outer circle represents one strand of the DNA, and the inner circle represents the other strand of the DNA. So what is happening here, only one of the strands is being modified. And that was very nice because we knew from other people's work that of the 2 or 3 type 3 enzymes that had been characterized, they were all single strand specific. And they get away with this because the orientation of the sites is selected for as it is important, and the 2 methylase subunit, the 2 restriction subunits have to be on the same strand pointing at one another before they'll cleave. And so this was very nice. And we've now done a large number of these, some cloned, some not cloned, and this turns out always to be the case with just one or two exceptions. There are a few systems out there that methylate both strands, but the vast majority, something like 98% of everything we've looked at, just recognizes a single strand. So we thought, well, if we can do this on cloned genes, maybe we can look at a whole genome. Let's take a whole bacterial genome and run it through the PacBio system and then see what we get. And so we took this organism, Chromohalobacter selexigens. It's a very relatively small genome. Using bioinformatics, we could find out where all the methylase genes were, and there were just 3 methylase genes on this chromosome. This one up here was a typical type 1 system. It's got an R subunit, M subunit, and S subunit, and we were expecting that it would produce a pattern like the other known type 1 systems in which there was 3 or 4 bases here, and then ends in the middle, and then another 3 or 4 bases over here. And sure enough, that showed up down here as that recognition sequence. Down here were 2 solitary methylase genes that didn't appear to have a restriction enzyme associated with them. There was no candidate gene next to it. And when we cloned these out and looked at them, and we discovered that this one, CSA1, recognized GATCY, RGATCY, shown here. We had predicted GATC, and so we were wrong in our prediction of that one. But there is no corresponding restriction gene, and because only about 76% of all of the sites were methylated, there couldn't be a restriction enzyme gene, because if there was, it would cut up the unmethylated sites. And so here we knew that this was a case where there was a solitary methylase. It's methylating these sequences for some reason, but we don't know why. The other gene, M.CSA3, was inactive in the genome. We couldn't detect any activity due to it, but when we cloned it, it recognized the sequence BA, where B means anything other than A. So it won't do runs of A, but if you have CA, TA, or GA, then it will methylate the A very nicely. And that turns out to be very interesting. It's encoded on a prophage. You know, bacteriophages, many of them, infect a cell, and then they integrate themselves into the genome. And as soon as they get integrated into the genome, the bacteria typically turns off all of the phage genes. And so what's happening here is you've got a bacteriophage that has a methylase that is promiscuous, does an awful lot of A residues. And so when the prophage excises, we imagine that the methylase genes get turned on, methylates all of the DNA in the phage, and so now it is pre-protected against restriction. So this is a way that phages have come up with a way to get over restriction systems. And we now know there are very many of these promiscuous methylases present on prophages. I will tell you about one other, but that we really have a lot of examples of this now. So after we did Chromobacter, we did 5 more. And when we looked using PacBio system and looked at all the recognition sequences and cloned everything out, I want to emphasize, we just didn't go at this point with a genome sequence. We cloned out all the genes to make sure that we were assigning the correct genes to the correct recognition sequences. We found a total of 27 methyltransferases in these 6 genomes, and 13 of these were brand new specificities, sequences that had never been seen before to be associated with restriction. So that was very nice. Then we looked at this E. coli strain that caused a massive break of enteric dysentery in Germany 2 or 3 years ago. And this is a strain that was of interest to a lot of people. Many, many people sequenced this strain, but we were the only ones who did the methylome. And the methylome turns out to be interesting. Don't worry that you can't read any of this, because there is a very simple message that I want to tell you here. This strain of E. coli here, you will see it has 4 methylase genes associated with it, and a gene down here that is associated with restricting methylated DNA. So many bacteria, turns out, have restriction enzymes that only cut methylated DNA, and this is the way they get around these prophages that have hopped out, put methyl groups everywhere, and now E. coli with this system in it can cut up those phage DNAs too. So this is just typical host parasite evolution taking place. But when you look at this German outbreak strain, it is absolutely loaded up with methylases. It's got a lot of restriction systems. A lot of pieces of DNA have been transferred into the strain, typical of many pathogens. But interestingly, these methyl-dependent restriction systems, of which there are 3 in E. coli, have all been lost. They're inactivated, they don't work, and so this allows all of these pieces of methylated DNA from phages or plasmids or wherever they come from to get into E. coli. And we've started to do a little bioinformatics analysis here, and we find that it is very common when you're looking at strains that are pathogenic that they have lost the ability to get rid of methylated phages. And so phages can then get in, bring in DNA, bring in toxins, bring in all sorts of things that can make strains pathogenic. And this might turn out to be quite a useful diagnostic marker if you're, say, looking at a population of bacteria, you're wondering if they're pathogenic, if they're about to become pathogenic. And this is a very nice surveillance tool. And we've been working with the FDA and the USDA to look at this in some more detail because they're very interested. Bacterial infections can be a big problem for them. There's also 1 or 2 very interesting genes, these first 2 up here. And like the one I just told you about, the methylase that recognizes BA, these methylases are also on a prophage, but they do every A residue. So even if you've got A, they will do all of them. And so these are really prolific methylases. It turns out that they're present in something like 100 other species that we have sequence data for in GenBank, and we've started to do a little work on them. This is what the Circos plots look like. So if I plot where all of the methylated A residues are on the 2 strands, they're essentially complete. If we do a sequence logo, we find there's just a little bit of something here, but essentially it's all A. And then this is when you look just at where the sequence plot shows you where the methylation takes place. It's just everywhere. So that was interesting. There was also a methylase that we found that turns out to be unusual, interesting, and I think may turn out to be quite important. In this case, what we've done is to do the Circos plot, and you can see there's a little bit of methylation on the outer strand and essentially nothing on the inner strand. This is encoded on a plasmid. We only found it when we cloned the gene out. And in Burkholderia, exactly the same thing on a plasmid, something that methylates one strand but not the other. And it's pretty promiscuous. It does all sorts of sequence, doesn't have any strong sequence content. And so you have to ask yourself, how could you methylate one strand and not the other? Well, the easiest way to do that is to think that the methylase is perhaps associated with the replication machinery. And so when you're making, say, the leading strand synthesis, if you've got the methylase going along, then it has the leading strand available to methylate, but can't easily get at the lagging strand. And so we've been thinking that maybe this is not really a methylase per se. The methylase activity is totally incidental, but perhaps it's taking advantage of the fact that it's a base flipper. And maybe what is happening is that this methylase is actually working essentially as a helicase to open up the DNA using base flipping, because I think if you look here, you can immediately see that as soon as you've got one base out, you've lost half the stacking interactions for the next base, and so it would be easy to flip that one out too. And so our speculation at the moment is that here we've got a case of something that used to be a methylase, but has now been diverted to become a helicase. And we have some preliminary evidence as a result of purifying this, that when we try to purify this out of E. coli that is expressing it, the DNA polymerase comes along with the methylase. The two seem to be co-purifying. So at the moment it's a hypothesis, but I think it's quite attractive. And in fact, this is another case where there are hundreds of these on plasmids when you look at GenBank sequences. So this graph is to show you what has happened as a result of PacBio sequencing system. This is the rate of discovery of type 1 and type 3 methyltransferases for which we know recognition sequences. And so you can see, starting back here in 1970, which was when the very first one was done, there was very little known, and then slowly some type 1s came along. Most of these type 1s came from restriction. There was a guy called Junichi Ryu came up with a very clever method of working out what recognition sequences were, but none of these were from cloned genes. This was just by straight biochemistry. And then in 2012, all of a sudden it took off. And this was because of PacBio. We started to look at a lot of genomes. We have now data for something like more than 400 genomes that have been done by various labs around the world. We've been collaborating with a lot of these. And the solid line shows you ones where we know the recognition sequence and we know which gene causes it. And the dotted lines are cases where we have the recognition sequences, but we don't know which genes cause it. So you can imagine, if you've got a bacterial genome, it has 2 type 1 systems in it, how are you going to decide, even if you've got 2 patterns, which does which? Sometimes we can make a good guess, but at the moment we don't have enough data to be sure that our guesses are correct. And we have to do a lot more cloning, a lot more biochemistry to be sure that our guesses are correct before we start assigning these. But you can see the rate of discovery here is just enormous, absolutely shooting way up. Along the way, we've also discovered an interesting type of type 1 system. Normally, they have the 3 genes that I told you about before, but we now have half a dozen examples where there are 1 S gene, 1 R gene, but 2 methylase genes. And what is going on here is that the recognition sequence has an A residue in one half of the recognition sequence, but the other half only has G and C. And typically, the methylation is such that it does 1 A here and then 1 A over here. Here, there is no A on this side, and so what's happening is it's actually N4-methyl-C is being used to protect. One of these methylases does the A, the other one does N4-methyl-C. So that's a new finding that just popped out from these. We're very interested in how these specificity subunits actually recognize the sequences that they look at. You can see this is sort of a general description of what the type 1 sequences look like. The variations that we see today, there are these 2 regions. One recognizes one half of the recognition sequence. This one recognizes the other half. Some have 3 TRDs, some only have 1 TRD. And these are all a little anomalous. They recognize different sequences. And interestingly, they're not all part of the same system. You don't find these genes next to one another all the time. Sometimes you find genes at very different parts of the chromosome interacting with one another. This has never been seen for type 2 or type 3 systems, but turns out it is quite prevalent in a lot of type 1 systems. And that makes it more difficult to work out what is interacting with what. And so we need some interesting bioinformatics puzzles there. The TRD sequence itself can be anywhere from 2 to 5 bases that it recognizes. We have some examples that recognize 5 specific bases. And this suggests that we might actually be able to mix and match these, because it turns out you can recombine them and they do very well. We could actually make a 10-base pair recognition sequence fairly easily. Maybe we could make bigger. And our feeling is that these systems, which are exquisitely well controlled, if we could learn how to engineer them, we might actually be able to make things that are even better than CRISPRs for doing gene engineering of one sort and another. Because one of the problems with CRISPRs and TALENs and zinc fingers is they have off-site activity. They go to places you don't want them to go. And this doesn't seem to be a problem so much here. The submotif sequences, you can have variations, you can have purine pyrimidine, you can have ACG and not T, and a variety of different things there. And then the number of Ns that separate the motifs can be anywhere from 4 to N. So there's quite a variation. And the variation is such that when we look at new systems today, there's a greater chance we'll find something that we've never seen before than that we find something that we have seen before. So we're constantly finding new recognition sequences here, much more than had ever been anticipated. So what have we learned? Well, we've learned that there are a lot of solitary methylases, things that don't have a restriction component. In some cases, we can be sure there's no restriction component because the methylation is not complete. In other cases, it is complete, but we just can't find any evidence for a restriction gene. A lot more variability in recognition sequences than we'd expected. The type 2 methylase specificity matches that of the restriction enzyme, and we don't know why that has been selected for, but it's interesting. There's an interesting puzzle there. And now we found that there are many, many new non-specific methyltransferases on prophages. So here's examples where evolution is really at work in getting prophages to evade host restriction systems. And I just want to make a pitch for PacBio sequencing. I don't own any stock. I have no interest in this company other than this is a wonderful method for sequencing DNA. First thing, of course, from my perspective, it's easy to determine methylase recognition sequences. There is no other way of doing that. The functional annotation of methylase genes now becomes much more straightforward, and we're learning how to match genes to recognition sequences, although we still have some way to go. Closing bacterial genomes has become almost trivial. One of the things about PacBio is that it can give you reads up to 20 kilobases. And so whereas Illumina and Ion Torrent just give you short reads, and making the overlaps and working out how to put a sequence together is quite difficult to a point where you usually can't do it. And with PacBio, you usually can do it. We have looked at probably 40 genomes, of which I think 35 we closed just by using software. No additional experiments needed. Whereas with these other methods, you need other things. The other thing is the accuracy of sequencing is enhanced. Turns out that while a single PacBio read is not terribly accurate, it has some variations in it, but those variations appear to be completely random from everything that's been looked at so far. And because you typically get somewhere between 50 and 100-fold coverage of every base along that sequence, it is incredibly accurate. And in fact, they've now been shown that there are quite a lot of human polymorphisms that were attributed as a result of Illumina or Ion Torrent or 454 sequencing that are simply wrong. They're actually just errors, systematic errors in the sequencing approach, and they have been corrected by PacBio. And there are a number of papers in the literature showing that. So it's a terrific method. It's a bit expensive. The machine is expensive. The runs are not terribly expensive. But the value that you get out of it, I can tell you, having a closed genome is so much more valuable. It's 100 times more valuable than just having a shotgun sequence set. So for that alone, it is really worth the extra expense. Finally, I just want to acknowledge the people who've been involved. I don't want to go through them all, but there are people at JGI. We're just starting to collaborate with the Sanger Center, who are going to sequence 3,000 genomes. From one of the big English culture collections. They're going to share all the methylase data. We're going to do all the analysis for them. And there's a bunch of other places, JGI, the Broad Institute, the Craig Venter Institute, and lots of others, FDA, USDA. A lot of people are doing this now, and I really appreciate it. They've been incredibly generous in sharing data. If they want it kept private, we keep it private. If they want it made public, then we make it public through Rebase. And I would thank all of you for your attention, and I'm happy to take any questions you might have.
So we have 10 minutes for discussion and questions. Yes, please.
So I was wondering about the error rates of this. How specific are they really, and do you have any areas?
Okay. So do you mean the error rates in determining methylase recognition sequences? So that is a little bit difficult to get at the moment for the simple reason that the motifs are defined by PacBio software. And we know that the method they're using is not absolutely perfect. They often come up with sequences that are not clean and cannot be absolutely correct. But we know how to then go and look at the original data and go back and check it, and we're actually writing programs at the moment to increase the calling of the recognition sequences. For the most part, though, they're very good. If you get 100-fold coverage, usually they give a very good job. But if you get down to sort of 20, 30-fold coverage, then they're often not quite so good. But it's a problem with the software. It's not a problem with the basic sequencing. Yeah, if you really have good coverage, let's say I get 100% coverage for a genome, then from everything we've done so far, the recognition sequences are 100% accurate. Yeah. Yeah.
Is there any enzyme to demethylate the bacterial DNA sequence so far found?
Say that again?
Any enzyme to demethylate the DNA sequence?
Okay, so let me talk a little bit about demethylation. So cytosine demethylation is a bit of a problem. You can do it using these TET enzymes. It's known that this is done. One of the problems with PacBio sequencing is that it does not give really good values for 5-methylcytosine unless you first oxidize 5-methylcytosine, either to hydroxy, formal, or carboxyl. If you oxidize it, then you get much cleaner signals. And the TET enzyme will do it, but it's very expensive. And we're actually working on an enzyme at Biolabs at the moment that comes from a different source that is very much better at doing that. And we hope that that will come into routine use within the next few months.
Okay.
But we've really focused on N6A and N4C at the moment.
Okay. And if there's some homolog of the methyltransferase in the higher organisms, such as eukaryote.
Right. So the specificity of, say, the human DNA methyltransferase is determined by factors other than the methyltransferase. So it is obviously looking at chromatin, it's looking at proteins bound to DNA, and these influence what can be methylated and what can't. There is no inherent specificity that we've been able to find in the eukaryotic methyltransferases.
Thank you.
Okay.
There's a lot of interest at the moment in using zinc finger nucleases for gene therapy, and I was wondering how different or similar they are in terms of their recognizing the DNA to these restriction.
Totally different. Totally different. The mechanisms are completely different. And the problem with the zinc finger nucleases is they have a lot of off-target effects. And in general, the longer the recognition sequence, the more likely you're going to get off-target effects, because it's difficult to recognize a really long sequence and maintain the accuracy all the way along the sequence. The homing endonucleases, I.SC1 and so on, are great examples of this. They have very good specificity at some point in the sequence, but then very low specificity elsewhere, and so you always get off-target effects with them. And that limits their use for genetic engineering, for gene therapy, and that kind of thing.
You also said, like, if you look at these restriction endonucleases and the methyltransferases, you can't see anything in the structure that would predict the sequence that it's recognizing.
Neither the sequence nor the structure.
So with the zinc finger nucleases, is there anything about their structure you can see?
Well, yes,
predict
because
what they would bind
you know
to?
the rules.
Okay.
So for the zinc finger, you know the rules, you can design them, but what you end up with still has problems. So, the problem lies in the way in which the protein is recognizing the DNA. And what's happened with many of the zinc fingers is that they evolved initially to recognize short sequences, and now you're trying to make them longer, and obviously we've not quite figured out how to do all the protein engineering properly so that you really get good specificity at every single base. It's a problem. There's a question. Yeah, we'll take you next. Okay.
Do we already know the mechanism of that timing-based methylation identification system?
Well, so you mean the timing when I was talking about cell timing and so on?
The DNA matching that certain point gets a longer time to match, that identification system.
Oh, you mean for the PacBio? Do we know?
Yeah.
No, we don't really know that. But it's sort of pretty easy to think. The way in which polymerases work is that as they go along copying a DNA strand, they look several bases ahead in order to get the next nucleotide lined up and ready to go in. I mean, this is how they achieve such good efficiency. And so obviously whatever is doing the probing ahead is sensitive to methylation in some way. But just how that works, it's not been worked on. This is all done using 529 polymerase. There's been a lot of work done on 529, but not at that level of detail yet. Okay, so there's one over here.
Sir, most of the methylation has been found in a promoter region, in the CPG islands.
Sorry, I missed that. Say that again.
The DNA methylation is more prominent in the CPG island, in the promoters.
In eukaryotes, yes.
Yes, sir.
Right.
So, is there any mechanism to recognize the only the C at present in the promoter, the region, as compared to the inside
Right, so
the gene?
it's clear that with eukaryotic methyltransferases, they prefer CPG, so they like to do CPG, but they also do CWG, so they do CAG or CTG, they will do that. And the specificity is not in the methyltransferase. We have a very good methyltransferase that recognizes all CGs. It comes from a spiroplasma. The SSS1 methylase, which does all CGs, it doesn't look at the sort of target recognition domain there, the region that recognizes the DNA sequence doesn't look anything like the eukaryotic methyltransferases. So the specificity is by other factors. It's not inherent in the methyltransferase in eukaryotes as far as we can tell.
Have you ever checked the methylation in other nucleotides other than the A and C?
Have we?
Have you ever checked the methylation in other nucleotides?
In higher eukaryotes?
Yes, sir.
Yeah, so if you look in humans, mice, or mammals, it's only M5C, so it's only 5-methyl-C. If you look at Tetrahymena, it has N6-methyl-A. And there are certainly other organisms. We've been looking at a few organisms that have come, marine organisms, because I also work with a group that are doing marine organisms. And there are funny bases that are present. So one of the nice things about the PacBio system is that it can recognize funny bases when they're present in DNA. And we've just started a big program to look at a lot of modified phages where there's maybe hydroxy-U or various other modifications that are around to see what sort of signals you can get. It can also recognize phosphorothioates, so there are some restriction systems that put a sulfur atom in the internucleotide phosphate. They replace one of the oxygens with sulfur. That leads to an altered signal with PacBio, but it's not a very good one. We don't know how to interpret it properly yet, but there's a lab at MIT, Peter Didon's lab, who is developing some better chemistry to allow you to look at that in much more detail. So there's a lot of modifications in DNA that I think we don't understand terribly well at the moment. They've pretty much all been found in phages, but we're pretty certain that they're in the genomes too. But, if you think about the way in which we sequence DNA, first thing we do is we clone it. And so if there was any modification, you lose it immediately. And so it's only when you go back and look at the un-PCR DNA, unamplified DNA, that you can hope to find these peculiar bases.
It was a really good talk. I was curious about this new technology that you were developing. And is the idea to use the endogenous DNA recognizing capabilities of restriction enzymes to kind of bring these two portions of the two enzymes together? Or are you going to use endogenous restriction enzymes to engineer your own site recognizing proteins?
Yeah, so my interest in engineering is all in the type 1 systems, where we would like to work out how to make better specificity subunits that have altered recognition sequences of one sort and another. But my interest here is really as a basic researcher, not because I'm trying to make tools that biolabs can sell you at great expense. Okay, I'm interested in the basic aspects of all of this because I think bacteria are incredibly important, and we're not spending enough money studying them. We cannot live without the bacteria that live in our guts and on our skin and in our mouths, and they have a tremendous impact on health that we don't know very much about at the moment. And I think one of the very important things we need to do from a health standpoint is understand much more about how these bacteria are working, what they're doing, how they can help us. In the discussion group yesterday, I talked about this wonderful example of Clostridium difficile, which is a very nasty hospital-acquired infection that a lot of people get these days. This organism is resistant to almost all antibiotics that we have, and there are some strains out there for which there is no antibiotic that will kill it. And yet, if you do something, sounds disgusting, called a fecal transplant, in which you take stool samples, you sort of try to filter out all the crap and just get the bacteria, and then feed it to someone with a C. difficile infection, often they are cured. And it's the only way we know to cure it. So that tells you that in your normal, healthy gut flora, there is something that can keep some of these very dangerous pathogens at bay. And we don't know how they do it, we don't know what's doing it, but this is something with really a lot of potential for healthcare. I mean, wonderful potential. And, you know, and bacteria are relatively simple. People are so goddamn complicated. But bacteria, nice and simple, they're lovely. I love them. Okay?
So if there is no more questions, so let's thanks again.
Thank you very much, Dr. Roberts and Professor Shiomi. And now, look at the flora on the stage. It is presented yesterday for this meeting by the HOPE alumnus who participated in third HOPE meeting. And now, next program is poster session. Please move to the next room, Matsuba. Today's presenters are them. And this blue. And after the poster session, lunch will be available in the restaurant Il Leone at the Grand Prince Hotel, New Takanawa. After lunch, we will take a short trip to Keck and NIMS for research facility visit. And please get on the bus, team A, B, C, D, get on the bus number 1. And team E, F, G, H, get on number 2. And I, J, team I, J, K, L, get on the bus number 3. And when you go to the restaurant, please bring your umbrella. And don't leave your umbrella at the restaurant, please. And if you have not umbrella, we prepare it. And so you can borrow the stuffs and please ask for us. And the bus will depart at 12:30 from the entrance of the Grand Prince Hotel, New Takanawa. Please make sure you come where the bus is by 12:20. The bus will start at 12:30 sharp. So please do not delay. And after coming back from the research facility visit, dinner will be available in the restaurant Il Leone at the Grand Prince Hotel, New Takanawa. Thank you very much.